All right. Good afternoon, everyone. I understand that Christian just wrapped up, which is great. So I'm going to get started here. My name is Gerard Honig. I'm a member of the Board of Trustees, as well as a member of the Scientific Advisory Council for the Peggy Lillis Foundation. And it's really a pleasure to be with you all today. I'm going to be going into presentation mode, so if anybody has any issues either seeing the presentation or me or hearing me, just let me know. Otherwise, we'll dive right in. Just to tell you a little bit about myself, I've been working with the Peggy Lillis Foundation since 2015. I am a research scientist by background. I've studied microbiome research and physiology and immunology, and I've been working in the field of gastroenterology and immunology for about 10 years. I am employed at the Crohn's and Colitis Foundation, which is another patient advocacy organization, where I am Associate Director of Research Innovation, and I run a variety of research programs. And really, the majority of my professional career have been focused on how we can advance basic research advances into concrete practical solutions for patients. And it's really a privilege to address this group again. I presented in 2017 on the same topic, and look forward to a great session. I hope everyone is comfortable and safe as possible under the circumstances, of course. I'm going to present a couple of slides and talk a little bit about the fundamentals of C. difficile infection, its biology, and its public health impact. If you have any questions during the presentation, if you could chat them in the box or hold them, we'll have plenty of time after the presentation for a robust discussion. And of course, many of the people on the line have personal experience and personal knowledge of C. difficile, which we look forward to your sharing. You're welcome to use the chat box for that. Bernie and I will monitor it. You're also welcome to click the participate button in order to speak, so we can hear and everyone can hear your question, or you can even use your video, and if you'd like as well. So with that, I'm going to dive right in. So the overview of the content of this talk. C. difficile bacteria are a type of bacteria that can reside in the intestines, as I'm sure many of you know. The gut microbiome is a community of beneficial bacteria that also lives in the intestine and has many important roles, including protection from infection. C. difficile infection is really caused by a combination of two factors. One is a disruption of the gut microbiome, typically by antibiotics, as well as exposure to C. difficile bacteria. C. difficile can spread readily in healthcare facilities as well as in the community, and incidence of C. difficile infection means that it's really a very urgent public health threat, as we all know, but there have been some recent improvements which are encouraging. So C. difficile as an organism is a type of bacteria. It belongs to the class Clostrides. It's also known as Clostridium difficile. So Clostridium difficile and Clostrides difficile, don't be confused. It's the same thing. It's really a semantic difference, a difference in nomenclature that was introduced fairly recently, but it refers to the same type of bacteria. So this is a bacteria which you can readily find in natural environments such as soil or swamps. And it especially grows in environments that are low in oxygen. It's also known as anaerobic. Really ancient form of bacteria. It's also importantly found in the feces and especially the intestinal content of many different types of animals, including humans, and certain types of Clostridium difficile, not all, but some, have the potential to produce toxins that can damage the intestinal lining, accounting for its importance as a pathogen. So to speak a little bit about Clostridium difficile, and its life cycle, because it's important for how it spreads. C. difficile bacteria can exist in multiple states. The vegetative cells, which are illustrated, actually, this is a photo taken from both of these are photos taken from on the upper left, So these are, you can think of them as cells that act like a growing plant. They can grow, they can move a little bit, they can also reproduce themselves. These vegetative cells have the capacity to adhere to the lining of the intestine. They can be killed by antibiotics. The vegetative cells can be killed by antibiotics. And these vegetative cells can also produce the toxins that cause diarrhea, and they can also produce spores. So what are spores? Spores you can think of really like the seeds of a plant. They are small, they're hardy, they're long-lasting, and essentially they can sit around for perhaps tens, maybe hundreds of years, and really waiting for the right environment. So they are generated when the vegetative bacteria feels itself to be under stress. For example, when an antibiotic is applied, typically spores are produced. And so it's a stress response that's storing the genetic material of the bacteria for the eventuality that in the future that spore will encounter a more favorable environment. So it has the capacity to sense the environment and germinate, just like a plant, germinate and form new vegetative cells when the conditions are right. So these spores, importantly, they can reside on surfaces, and they're in fact very difficult to kill in surfaces. Typical sanitizing products like alcohol don't work particularly well, and you need to use bleach or ultraviolet light or other more intense disinfectant protocols. And importantly, however, and they can also reside in the gut and stay in the gut, and when they're in the gut, they are also not sensitive to antibiotics. And so spores are really this like very hardy, resistant seed of the bacteria. So now let's transition to discussion of the microbiome, which is the environment that these cells live in. So the microbiome is a balanced community of bacteria that are essential for human health. And these bacteria live in different surfaces within the body, on the skin, within the respiratory system, but especially within the gastrointestinal system. And these microbiome bacteria play a lot of important roles in human health, especially defense against infections in all of these locations. And here you see illustrated just a few of the many different types of bacteria that can reside in the stomach, in the skin, and in the lower gastrointestinal tract. So these bacteria are really a critical part of our bodies and our physiology as human beings. Animals have co-evolved, really all animals that have an intestine have co-evolved with microbiome bacteria for millions of years now. And in a healthy individual, microbiome bacteria can live in balance with each other and also with the human immune system. And in fact, these bacteria accomplish many important roles within the human body, including digesting certain nutrients that we are incapable of digesting without the help of these bacteria, generating a lot of the energy from starches, for example, that we eat. And they also can basically teach our immune system and train our immune system in terms of how to recognize bacteria and differentiate helpful bacteria from harmful bacteria. And so these are really important roles, but perhaps the most important function is preventing other dangerous bacteria from invading these spaces within the body and gastrointestinal tract. So the bottom line is that individuals that have a healthy, balanced microbiome typically can be exposed to C. difficile bacteria without experiencing a C. difficile infection. And in fact, they can even host living C. difficile bacteria within the GI tract without those bacteria getting out of control. So how do these bacteria then lead to a Clostridium difficile infection? Because we need to make a distinction between Clostridium difficile bacteria and Clostridium difficile infection. A person can have Clostridium bacteria, but would not have the diarrheal symptoms that characterize the infection. So infection is the presence of the bacteria plus the symptoms, as opposed to the bacteria itself. So how does this happen? It typically is happening with antibiotics. So illustrated here is a section of the intestinal lumen with a representative range of many different types of bacteria that you can typically find within the intestine.
Gerard, can you?
Now, yes,
Hey, it's Christian. So people can't. Your slides are not moving.
they're not moving. Okay, how's that? Okay, is that better?
Yes, that looks a lot better.
Okay. All right. Okay. All right. Sorry about that. Okay. So just so you guys can see the pretty pictures, I will quickly run through them, but I won't go through the content again. Here's an electron micrograph of Clostridium difficile, what the vegetative cells look like, and on the top left, and the spores on the bottom left. Picture of the human body with just a few of the different types of bacteria that are present. And finally, this lovely cartoon that was published in the New York Times in 2014. Okay, moving on. So what happens in the human microbiome is that it can be susceptible to depletion by antibiotics. So of course, antibiotics are critically important for human health and have really dramatically changed life expectancy within human societies by allowing us to deal with infections that in the past would have run out of control, would have gotten out of control and led to lethality. But there is a harm associated with antimicrobials, as many of you know, which is that the diverse microbiome, which is present within the intestine, as shown on the left-hand side here, can become depleted. So when you can take an antibiotic for a lung infection, a skin infection, or even for a gastrointestinal infection, you can kill the bacteria that are causing the infection, but you're also killing a broad range of different bacteria, healthy, beneficial bacteria. And this creates an opportunity for dangerous bacteria that are really just lying around waiting for this kind of environment to expand. And those bacteria can include Clostridium difficile, but there are many others, as well as viruses that can also take advantage of this environment, such as may be the case with COVID. So the expansion of these bacteria that particularly thrive within this deranged environment can lead to Clostridium difficile infection, as well as other types of enteric infections. But we also know that the microbiome, like many parts of the body, has the capacity to heal itself and repair itself over time. So that many people can actually recover the healthy microbiome even after recovery from antibiotic exposure or even Clostridium difficile infection. So how does C. difficile infection actually cause diarrhea, which, as we know, is the kind of key symptom of overt Clostridium difficile infection? So that is summarized here. So as we discussed, under normal circumstances, the gut lumen, which is the inside of the gastrointestinal tract, which is at the top here, and the layer within the middle is actually the intestinal epithelial layer, the wall between the rest of the body and the inside of the GI tract. And whereas the pink represents bacteria. So there's a density of bacteria within and the inside of the gastrointestinal tract. Antibiotics kill many of these beneficial bacteria, allowing Clostridium difficile to gain a foothold within this environment. Now, Clostridium difficile is then essentially activated and takes advantage of its new position of dominance to create conditions that it likes, but other bacteria and humans don't. And it does this by secreting toxins. And so toxins are molecules that can actually damage the intestinal wall within, particularly within the colon. And as a result of this damage, you can have bacteria that can leak actually into the body, which is which really puts off all kinds of alarm bells within the immune system. And you have infiltration of cells from the blood that are designed to deal with inflammation, but can also are going to cause leakage of blood into the gastron, into the stool, and damage to the wall, and diarrhea, which, if left unchecked, can spiral out of control and cause an uncontrolled inflammatory reaction, which can lead to conditions like toxic megacolon, or even shock and sepsis, which can be fatal. So C. difficile infection fundamentally is a 1-2 punch, where diverse gut bacteria can, under normal circumstances, can protect us from infection, whereas the antibiotic depleted microbiome leaves open a vulnerability. So, and C. difficile, which C. difficile can take advantage of. So you really need the combination of two different things. One is C. difficile exposure, which is necessary but not sufficient in and of itself. And the second is disruption to the microbiome, typically caused by antibiotics. And those two things together can lead to clinical infection. So the symptoms and risk factors, many of you, I'm sure, are familiar with, but just to review, symptoms can develop within a few days after antibiotic administration, and can include diarrhea, which are defined as loose or watery stools and frequent bowel movements over several days, fever, stomach tenderness or pain, loss of appetite, and nausea. And in severe cases, this can progress, as we heard tragically in the case of Peggy Lillis, this can progress to an out of control inflammatory reaction, and shock, and even death. Factors that increase the risk of C. difficile infection include antibiotic administration, being 65 or older, recent residence in hospitals or other types of healthcare facilities, particularly nursing homes, a weakened immune system, such as might be the case for a patient who is recovering from cancer therapy, or who suffers from autoimmune diseases, known exposure to C. difficile, and especially previous infection with C. difficile. But it's very important to mention that, with the exception of exposure to C. difficile, none of these factors is required for C. difficile infection. So there are instances where people can get C. difficile infection in the absence of antibiotic administration, although it's unusual. Certainly, it can happen in younger people. There certainly has been an increasing trend of incidence within people contracting C. difficile within the community with little or no exposure to healthcare facilities. And certainly, people that don't have autoimmune diseases can have C. difficile, and of course, prior experience with the infection is not required. Now let's talk about recurrent C. difficile. So how exactly does this happen? So C. difficile is a bacterial infection, and there are certain antibiotics that can kill the vegetative forms of the bacteria. They don't kill the spore form, so that's why C. difficile can be said to be in some ways resistant to antibiotics. But still, antibiotics typically are effective most of the time at suppressing the vegetative cells and suppressing the active infection if it's caught in time. So you take the antibiotics, and then the diarrhea can subside. However, the problem that can occur is that the antibiotic treatment itself, that is being given to kill the vegetative cells, can actually worsen and further damage the microbiome. So the treatment can actually increase the longer-term risk in the future of C. difficile. So the C. difficile spores also can persist within the gut, and they can also spread easily. For example, the patient's house or hospital room may become contaminated with spores that are very difficult to disinfect. And so patients can get reinfected, or perhaps they may need to take antibiotics for another reason later. And so basically you can enter into this vicious cycle where the diarrhea and the active infection requires more antibiotics, which also increases the risk of the infection recurring. So this can become really a very intractable situation. And if there's time and interest, we can talk about some of the emerging concepts in terms of interrupting the cycle by restoring the health of the microbiome. Okay, second. Right. Okay. So C. difficile has a really important public health impact and has for many years. So C. difficile causes close to 500,000 illnesses each year just within the United States, estimated. About 1 in 6 patients who contract C. diff will contract it again within the subsequent 2 to 8 weeks, and more people are most likely can contract it within a longer time frame after the initial infection. And importantly, C. diff can be deadly, as we discussed. So, for example, in this study cited by the Centers for Disease Control, 1 in 11 people over 65 diagnosed with C. difficile infection died within 1 month of the initial infection. So C. difficile infection is an extremely common cause of infectious diarrhea. It's a major burden on the healthcare system. It's one of the most common hospital-acquired infections. It frequently recurs and kills thousands of people each year. And we really have a lot of the knowledge in this area has been generated by a wonderful team of researchers at the CDC, one of whom we'll hear from, I believe, tomorrow. So how does C. difficile spread? Like this is the case for many different infections, and Clostridium difficile probably the most common. Here you have an individual that takes antibiotics, and it creates this opportunity for the pathogenic bacteria to expand. And then different things can happen. Let's say, for example, the individual stays at home, stays in the general community. This can account for community spread, for example, within the home or in other settings. Alternatively, if the individual is residing in a healthcare facility, that can get spread readily within the healthcare facility, especially if there's inadequate sanitation in terms of hand washing and cleaning up the facility. And also, as we discussed, the bacteria can spread, especially the bacterial spores can spread in surfaces. So let's talk about the prevalence and burdens. We talked about some of the numbers. Now, Clostridium difficile infection, if you talk to individuals that have been working in the infectious diseases field for some time, historically it was considered, although people have been really known about antibiotic-associated diarrhea and some of the negative consequences for the long time. It really wasn't considered to be the level of public health threat that it is today until the early 2000s. And so here you see on the left-hand side a graph showing the number of hospitalizations rising dramatically between 1993 and 2010, particularly from the late 90s onward. And this had to do with the emergence of new and more virulent strains, more dangerous strains of the bacteria, which were first documented in North America in the early 2000s and led to, it was one of several factors that led to a significant increase. Now, we've actually seen a leveling off and even a moderate decrease in the number of C. difficile infections. I'm happy to report this is quite a different picture than when I first presented this in 2017. And so we're really starting to see pretty significant decreases, particularly in the healthcare-associated infections. These are infections that people contract while exposed to the pathogen at a healthcare facility. But it nevertheless remains the case that C. difficile infection remains an urgent public health threat, with hundreds and hundreds of thousands of hospitalizations every year, as well as thousands of deaths. And we also, and while there's been some progress on the healthcare associated side, due, we believe, to improvements in how antibiotics are used and how Clostridium difficile is handled within those facilities, we have not seen decreases in community-acquired C. difficile, which remains a common threat. So really what we've seen with Clostridium difficile, and we've seen it as Christian mentioned, with other infectious disease threats, is a transition where organisms that previously had not been recognized as important health threats can really emerge. They can take advantage of the way in which we live, the way in which our healthcare facilities operate, and really become dramatically important public health threats. So with that, I see we have a couple of questions, which is great. Are we able to have people join and ask their questions in person? If not, I'll start going down the list. And thank you all so much for your attention to address a few of these great questions. So Ronnie Valenti asks, does difficile also destroy the gut microbiome? Okay, so this is a great question. Okay, so difficile was developed in order to be a specific antimicrobial against C. difficile that cause less damage to the gut microbiome than previously developed antibiotics. And it was really selected and developed specifically for that purpose. So shorter answer is that difficile is in fact more targeted and more specific than other antimicrobials, including vancomycin, for example, which is quite broad spectrum. But it still does most likely cause some damage to the microbiome. It's believed that the relative decrease in the occurrence of recurrent C. difficile after treatment with the initial antimicrobial is likely due to the relatively more targeted spectrum. Kill spectrum is what it's called for difficile. Okay, now I'm back. All right, great. So I'll just keep going through the questions here.
Good, thank you.
While we're oh, great, wonderful.
Hey, Angela.
Hi.
Hello, nice to meet you.
You too.
Do you have a question or a comment?
I actually do. You were just talking about difficile. How about the probiotics like Floristore? Is that going to replenish? How well does things like that work?
Okay, so probiotics like Floristore are live bacteria that are intended to, as you said, the original concept was that they would replenish the gut bacteria. However, for the most part, the kind of bacteria that are present in commercially available probiotics are quite different from the dominant bacteria, the healthy bacteria that are present within the gut. So probiotics do have some benefits, especially for some people, not everyone, in terms of helping to resolve a variety of gastrointestinal issues. So the best advice is that you should that people can try them, and if they work for them, then that's great. But they appear to have a relatively modest effect on Clostridium difficile associated diarrhea and on antibiotic associated diarrhea more generally, probably because they're not necessarily quite the right bacteria. So they can have some modest degree of benefit, but they're unlikely to be a lasting solution for most people. Does that answer your question?
Yes, thank you.
Okay, good. All right, let's see. Does C. difficile have a role as part of the normal microbiota? This is a great question. The answer is that we're not entirely sure. So somewhere between 2 and 5% of the healthy population has Clostridium difficile in the gastrointestinal tract. And as we discussed, in most of those people, it never really leads to any problem. However, we don't really know whether it's playing any beneficial role, but it may. So, and the evidence for that is from a series of studies from Dr. Dale Gerning and colleagues, where they looked at the presence of close cousins to Clostridium difficile that are very similar to the pathogen, but are not able to make the toxin. And in general, people that have these bacteria that are very, very similar, they really are Clostridium difficile, but they're a close cousin that doesn't produce the toxin, are likely to actually be protected, suggesting that the presence of these bacteria can actually be beneficial, perhaps because they're occupying an ecological niche that the pathogen would otherwise be occupying. And in fact, Dale Gerting and his colleagues took this to the next level and have actually completed a quite interesting clinical trial where they administer Clostridium difficile that don't produce toxin and have, which results in some protection against Clostridium difficile. So it's likely that if not Clostridium difficile itself, some close cousins are in fact can be a healthy member of the gut microbiome. Why do antibiotics affect some people in causing C. diff and some people do not? So, okay, so there's a couple of different reasons why this might be. One is that somebody might or might not have C. diff or might not be exposed to C. diff prior to or around the time they're taking the antibiotics. So if somebody does not have C. diff and is not exposed to C. diff and takes the antibiotic, they might experience other issues related to antibiotics, such as the more common antibiotic-associated diarrhea. But in the absence of the bacteria, they're not going to experience C. diff. However, even in people that are exposed to C. diff and take the antibiotics, some will progress to Clostridium difficile diarrhea and some will not. And the answer why it is that some will and some won't, it's not entirely clear. It probably depends on the composition of that person's microbiome and the interplay between the antibiotics and that person's own natural microbiome. Because microbiome, the interesting thing about it is that it can vary a great deal from person to person. And we don't really understand fully the nature of that effect. But there's some kind of tipping point where the damage to the microbiome has to progress past a certain point, and if it doesn't, the microbiome can reestablish itself and become more healthy. And in other cases, it can't. And why that is, and what are the factors that cause that are incompletely understood, but are believed to be related to differences that those people have prior to taking the antibiotics in their own gut microbiome. Okay, so do we have any results from the poll that we can review? Perhaps I'll keep going through the questions, but Bernie or Christian, if you're able to access those, that would be great. We can read them. If not, we'll just report them later. Okay. Let's see. How does C. diff interact with chronic illnesses like gastroparesis? So the relationship between C. difficile and chronic illnesses is that in a proportion of patients, a small but significant proportion of patients that experience C. difficile infection, that can trigger long-term gastrointestinal disturbances, including gastroparesis or post-infectious irritable bowel syndrome. And those things sometimes can establish themselves and perpetuate themselves even in the absence of recurrence of the infection itself. And so that's been well recognized potential effect. As far as susceptibility in general, it's not believed that patients with gastroparesis would necessarily have a higher risk of contracting it in the first place, but there are certain chronic gastrointestinal diseases, particularly inflammatory bowel diseases, that are associated with significantly increased risk. And typically these are diseases that are that are in the category of autoinflammatory or autoimmune diseases with a GI manifestation, which unfortunately are quite common. Okay. What is the best way to restore the microbiome post C. diff? And how do you distinguish between C. diff infection and a stomach bug? Those are great questions. So I'll start with the second, which is a little bit simpler. Generally speaking, the ways in which C. diff is recognized specifically are to isolate the stool, isolate the different bacteria growing in the stool and grow them, and then identify them under a microscope. That's one way. And also growing the bacteria under conditions that we know that Clostridium difficile likes versus other types of bacteria have other conditions that they like. So that's one way. Another way is to analyze genetically the composition of the stool in order to see whether Clostridium difficile is present and other bacteria are present. And so there's a variety of laboratory tests that can specifically identify Clostridium difficile within a stool sample. And that's generally how that is distinguished. However, it is true that sometimes you can have multiple different types of bacteria present that could be causing the diarrhea. So for example, in children, we know that a very large proportion of infants have Clostridium difficile in the stool. But very few of them actually are experiencing diarrhea. And in fact, most of the time when those infants are experiencing diarrhea, it's typically caused by another bacteria. And so this is kind of is a well-known challenge, but we do have some tools to address it. What is the best way to restore the microbiome post C. diff? So there's a couple of different ways in which this has been explored. One way, which is gotten a lot of attention recently, is with a fecal microbiome transplant. So this is the basic concept of the fecal microbiome transplant, is to transplant into a patient who has recurrent C. diff the gut bacteria that are represented as from a healthy person. So that's one way. Another way, which is kind of the next generation of microbiome transplantation, is to identify specific defined bacteria within the microbiome that are responsible for the benefits. Perhaps it's 13, perhaps it's 40, perhaps it's 7, and to cultivate them and test those in isolation in order to see whether that can be helpful. And that's also being explored. And then finally, most likely for most people, however, a great way to maintain your microbiome health is to maintain your general health and to have a healthy diet, because the bacteria are eating what you eat. So eating a diet that's as high in fiber and vegetables, and generally maintaining your overall health and also avoiding unnecessary antibiotics and unnecessary exposure to healthcare facilities is probably the best way to maintain microbiome health. Are spores resistant to UV? I believe actually that ultraviolet is quite effective against spores. And so how can we improve cleanliness in hospitals? Well, a lot of this has to do with advocacy. So, we all need to work together in order to ensure that our hospital environments, as well as nursing homes and other healthcare facilities, are safe as possible. And that includes cleaning with bleach. Ultraviolet disinfection also can have an important role as well. Is there transmission of spores between animals and humans? The short answer is probably yes. So C. difficile can exist in a variety of animals, particularly animals that can that are used for meat production. However, the dominant vector, the dominant way in which C. difficile can be transmitted between humans and animals is probably actually through contaminated food products. So these are food products, either meat or vegetables that become contaminated by C. difficile, probably C. difficile that's being shed by farm animals. And this is very likely to be one of the factors in the spread of C. difficile within the community settings, as has been, as has been stubbornly remaining quite high despite the progress that we've seen in healthcare facilities. Is Difficid more effective than Vanco even after the use of Vanco? We don't know. So this is one of the main challenges, that Difficid was shown to be equally effective as vancomycin, but with a lower propensity for recurrent infection in the initial very important clinical trial that was performed. So it basically works just as well as vancomycin for the acute infection, but because presumably there's less damage to the microbiome overall, there is less tendency for those patients to have recurrent C. difficile afterwards, about 50% reduction. So that was great to see, but it was only evaluated in first-time C. difficile. So if you take a patient and you have the option of treating that patient with vancomycin or Difficid, then the Difficid was equally effective initially, but then had this fall off in terms of the reduced propensity for recurrent C. difficile. Unfortunately, the way that Difficid is often used in the clinic is that it's used only when the patient has already had recurrent C. difficile, which is fine. It's a reasonable thing to do. It certainly there shouldn't be any harm of that approach, but it's not clear that it has any long-term benefit at that point when the microbiome is already quite disrupted. And I guess we have time for one more question, I think, before we move to the networking and the expo. Do you have any recommended protocol for cleaning and disinfecting rooms and facilities for C. diff? So typically the best practices are thorough cleaning with bleach, and then there are a few other modalities that we talked about, including ultraviolet light, which can also be effective. And then the other important thing is isolation. So making sure that people that do have a confirmed C. difficile infection are given a private space, and there's with minimal risk of infecting other patients. Okay. So I think everyone is welcome to join the expo.